Good morning, everyone. My name is Titas Grabauskas. I'm an MD PhD student in Dr. Dugu Uchar's lab at the Jackson Laboratory, and today I'll tell you about my project titled Increased Frequency and CMV-specific Clonal Expansion in GZMK positive CD8 T cells. So one of the big things we're interested in our lab is understanding and quantifying inter-individual immune heterogeneity. And when it comes to this person-to-person immune differences, there's always this nature versus nurture question. Are they driven by genetics or environment? In 2015, Mark Davis's group built an impressively large cohort to answer this question of monozygotic twins, where each twin pair had identical genetic backgrounds but different environments, especially as they aged. And using this cohort, they were able to show that environmental factors are actually major determinants of immune heterogeneity in humans. Of course, the authors wanted to take this one step further and understand what kind of environmental factors are driving this heterogeneity. And it turns out that CMV is a key environmental factor driving it. So to illustrate this, here they show a figure with cell frequencies. So each dot here is a different cell frequency in the blood. And what they're doing is on the y-axis, they are showing the correlation between monozygotic twin pairs that are discordant for CMV status. So one twin is CMV positive, the other one is CMV negative. Was on the x-axis are CMV coordinate CMV negative twin pairs. And so if you expected CMV to play no role in immune heterogeneity, you would expect all the cell types to kind of line up right here on this diagonal line. However, as you can see, the distribution is much different, and a lot of cell types are falling away from this line. So therefore, CMV is a key environmental factor driving immune divergence. So what is CMV? Well, CMV is a DNA herpesvirus that establishes latency. It has very high prevalence, and according to the CDC, around 50% of adults are infected by age of 40 in the US. And most importantly, CMV has been linked to altered immune function and composition. So when it comes to clinical implications, CMV seropositivity has been associated with worse outcomes of tuberculosis and SARS-CoV-2 infections. On the other hand, very interestingly, CMV seropositive patients, CMV seropositive metastatic melanoma patients have been recently shown to respond better and have overall better survival following anti-PD-1 checkpoint blockade. 10 years ago, also Mark Davis's group also showed that healthy young CMV seropositive adults tended to respond better to vaccine responsiveness. So looking at the negative and the positive associations, I think it is fair to say that we don't completely understand what CMV does to our immune system. And so additional characterization efforts are needed. But before I go into my data, I just wanted to quickly show you what others have done to understand CMV immune signatures. So for example, in this paper from 2016, the authors took the CD8 T cell compartment, and using CCR7 and CD45RO, they were able to segregate the T cell lineage into 4 major subsets. So here are the naive T cells, and then once these naive T cells become exposed to an antigen, they start proliferating and they become activated and start proliferating and differentiating into central memory cells, into effector memory cells, and ultimately with this continuous stimulation, they will become CCR7 negative, CD45RA positive, termed terminal effector or terminally differentiated CD8 T cells. Now, when you compare right here in orange, when you compare these TEMRAs in CMV positive versus CMV negative donors, you can see that there is a significant expansion of CD8 TEMRAs in the blood of CMV positive donors. And this has been very well described across many different groups over the past decades in the CD4 and the T cell and the CD8 T cell compartment. And therefore, now expansion of TEMRAs in CD4 and CD8 T cells is now kind of considered to be a hallmark of CMV seropositivity. But of course, we understand that the blood is rich with immune cells, not other than T cells as well. So to that end, the overall study objective that we set out to conduct was a systematic evaluation of CMV linked immune signatures. So while the CD8 and CD4 T cells have been studied pretty well at the protein level, we wanted to simultaneously look at the B cells, the monocytes, dendritic cells, and natural killer cells using single cell technology. So to our study design right here, we compiled a huge data set with 6 independent cohorts, 370 donors, and these cohorts have been profiled using RNA sequencing, flow cytometry, and TCR sequencing. Right here, I wanted to show you the age distribution of our cohorts. So as an aging lab, we're mostly interested in older adult cohorts. However, we made sure to include some publicly available younger adult cohorts in order to validate or compare the signatures in young versus older adults. So, and the way we have structured the study and this presentation, essentially what we did is we used cohort 1 and cohort 5, which we have built in-house. These are newly built cohorts that we used as discovery cohorts, if you will, to kind of derive the transcriptional signatures and TCR signatures respectively. And then we used the rest of the cohorts to kind of cross-validate our observations. So to show you a single cell data from cohort 1, which included 145,000 cells and 27 donors, here you can see the major PBMC lineage that I've labeled, and I've spent a lot of time looking closely into each and one of these lineages and understanding if there are any frequency or alterations between CMV positive and CMV negative donors. And just to show you a summary of that analysis, as expected, we saw a lot of differences in the CD8 T cell compartment. However, our systematic approach also, I think, paid off because we at the same time we noticed within the same cohorts we noticed we observed differences in the NK cells, in the B cell lineage, and the PDCs. Again, it is important for me to emphasize and remind you that like while this is a summary slide from cohort 1, we have validated each and every single one of these observations across other cohorts, therefore giving us great confidence in these immune signatures. But at the end of the day, the CD8 T cell lineage did yield the most alterations in frequency. And so when we're looking at this lineage, and studying it, one specific subset that really caught our attention was the GZMK positive CD8 T cell subset. The reason why it caught our attention is because recently in high impact papers, this population, both mice and humans, has been shown to be linked to aging and inflammaging. So to illustrate that, in 2021, Max Artemov's group published a paper in Immunity titled "Comprehensive Profiling of Aging Immune System Reveals GZMK positive CD8 T cells as a Conserved Hallmark of Aging." So in this paper, they did many things, but right here they're looking at lean, healthy males, and they profiled their PBMCs using RNA sequencing, and then they split their cohort into young and older individuals, and they showed that the GZMK CD8 T cells were increased in frequency in the older adults. However, they never really looked at the CMV serostatus, making us wonder whether their observations are potentially confounded. So now going back to our data, this is here I've reclustered the CD8 T cells from cohort 1 and I've labeled them into 7 distinct subsets. So here are the naive cells, there is the central memory, effector memory, and TEMRAs. And when you color code this UMAP for CMV status, blue being CMV negative and orange being cells coming from CMV positive donors, you can see that the UMAP tends to get a lot more orange as the cells are differentiating and indicating an expansion in frequency. And now we're interested in this GZMK subset, which is right here. And what I did next is I just want to show you the quantification of the frequency of the subset at the donor level in this age-matched cohort, right? So you can see that indeed the GZMK subset is increased in frequency. And because we have multiple cohorts, I get to cross-validate these results in a younger adult cohort, 25 to 65-year-olds. Again, the orange and the CMV positives and the CMV negatives here are age-matched, showing the same significant finding. And another cohort 4, again, showing an expansion or increase in frequency in the GZMK positive CD8 T cells. And while this is very exciting and cool, we wondered whether we potentially implicating the subset with the CMV seropositivity in addition to aging and inflammaging. We wondered whether we could come up with more evidence to support this. So the next question that we asked was, well, is the increase in GZMK CD8 T cell frequency driven by CMV-specific clonal expansion. So up until now, whenever we're looking at the CD8 T cells, we're looking at a mixed bag of antigen specificity. So some of those cells might be specific to CMV, some of those might be to COVID, SARS-CoV-2, so on and so forth. And we know that when a CMV-specific T cell encounters a CMV-infected cell, it will recognize that using MHC TCR binding, and the TCR through the TCR activation, the cell will start releasing cytokines and perforins to kill the cell, but additionally will start proliferating and expand clonally. And this is, and specifically we wanted to quantify this clonal expansion using TCR repertoire diversity, which is its most simple form is the total cells over total clone is a ratio of clones to cells. And of course we use the our favorite package to do so, Alakazam, developed by the organizers of this meeting. And the reason why we picked the package is it's quite user friendly. It allows for a bootstrapping approach. It allows us to calculate the diversity using different Hill numbers, giving more weight to expanded clonotypes and so forth. So it's at this point, our hypothesis was that in CMV positive donors, you will have a CMV specific clonal expansion because of all this going on right here. And because of that, the overall TCR diversity is going to decrease. So to test this hypothesis, we turned to cohort 5, which is an older adult cohort as well, but it's profiled using 5' RNA sequencing. So here we have the RNA and the TCR data within the same cell. And this cohort includes 14 CMV negative and 6 CMV positive older adults. So first, what we did, we calculated exponent of Shannon's entropy to show within the memory TCR compartment, within the memory T cell compartment, to show that the memory TCR diversity is lower in the CMV positive donors. But of course, we have the single cell data, we have and so we can break this down even further to show that CD4 and CD8 TEMRAs, which was kind of expected, have lower diversity in CMV positive donors. But again, we saw this GZMK CD8 T cell subset come up here again, which was great. But at the same time, what we're doing right here is we're quantifying the overall TCR diversity. So we wanted to be a little bit more specific. So we know that TCRs are highly specific to the peptides that are recognized, right? And so we took advantage of recently published CMV eco cluster, which includes 26,000 TCR beta chains that have been linked to CMV. And so matching this data set with our TCR data, we found 500 cells in our data set that were putatively CMV specific, and this corresponded to 115 clones. Then we mapped that information back to our labels. And here we're quantifying the CMV-specific cell counts on the y-axis in the CMV negative and CMV positive donors. And again, the same populations that showed decrease in diversity come up here again. And most importantly, the GZMK positive CD8 T cell subset also pops up again, showing a 4.4 clonal expansion, CMV-specific clonal expansion, which I think is strong evidence indicating that its potential direct role in CMV latency control. So with that, we have undergone a systematic study of CMV-linked immune signatures uncovering broad PBMC compositional remodeling. I didn't have time to show you the whole study, but we verified previously known signatures, and most importantly, we found some novel ones and ones that have been previously linked to aging. So, for example, increase in frequency of atypical B cells, and of course the GZMK subset. So the GZMK subset we think is linked to CMV, and we show this using the increase in frequency, reduced TCR diversity, and CMV-specific clonal expansion. And so, interestingly, in 2023, recently, the Michael Brenner's group published a very nice paper in Nature, showing that GZMK, unlike other granzymes, is able to cleave complement components in order to activate complement cascade. And we all know that complement cascades are huge, are inflammatory, are pro-inflammatory, right? So we wonder now whether the previous associations of the subset to aging and inflammaging that I showed you earlier are potentially driven by CMV and this GZMK subset. Because of all this work, we have become strong advocates of reporting CMV serostatus as a standardized practice in immune studies. And if you have a single cell object, a single cell transcriptomic data set that you have not profiled your CMV, you have not profiled for CMV serostatus, I would highly encourage you to take a look at our preprint, but also visit our GitHub page where we have developed an accurate machine learning tool that can use that transcriptomic data to infer CMV serostatus very accurately. With that, I would like to thank a long list of mentors in my lab, our collaborators, funding, and of course, the research volunteers. And with that, I'll take some questions. Thank you.